next section, we're going to talk about lab processes. So in a previous visit, I talked about the importance of making sure frozen samples are sent with enough dry ice that'll be sufficient for when they arrive on the site of the lab, the central lab, that they will still be frozen. So we already kind of touched on that. But let's touch on the processing of labs. We need to make sure that we follow the lab manual. Some labs, when you draw, they want you to invert it. Some say 5 times, some says 10 times. It is important that you invert because the solution that's already prepared in that lab tube, they want to make sure that it's mixed within that blood that you receive from that participant. If they said protect from light, immediately, usually this is a green top tube, immediately after you draw that lab, you want to have a paper towel with a rubber band over it, or some type of cloth over it. Whatever, your site might have some type of pre-sleeve that they can put it in. But you want to make sure that you protect it from light for the period of time in which they indicate. Some lab manuals will have you spin down a specimen, but they don't want you to pipette it into a aliquot tube. They want you to send it refrigerated as is. So it just depends on how that lab wants you to process it. If you don't have what's called a refrigerated centrifuge, because a lot of sites don't, they just have the ambient centrifuge. Oftentimes the lab manual will already specify what you can do in that situation, and that's typically what's called an ice bath. So you will put ice in a container, add water, usually midway, and then you will sit those tubes in that ice bath for the period of time that the lab manual indicated. It's typically 10 minutes, but again, pay attention to the lab manual. And then you can put it into your ambient centrifuge, and you can spin it down and pipette it into the aliquot tubes accordingly. Sometimes you don't have the opportunity to ship same day, and so it might be some already indications of what to do in the lab manual when it happens. Usually ambient specimens can be placed in the refrigerator up to 42 hours before until you have the opportunity to ship them off. But again, I'm speaking from experience. Read your lab manual. So before you contact your CRA asking, what if I do if I don't have this, if I don't do that? Typically it's in your lab manual. Then if it's not in your lab manual, you want to contact the central lab. Usually it says investigator support or something like that. And always attach your CRA, your monitor, so they can be in the loop. And they may have a response to you quicker than the lab does. But you always want to make sure that you keep your CRA in the loop.